I spent a couple of weeks at Imperial College in London, in the UK, to work with Ilaria Dorigatti, a senior lecturer whose research focuses mostly on developing mathematical models to investigate mosquito-borne pathogens and their transmission dynamics, a topic which I'm also very interested in. There, I had the opportunity to present a seminar showing the main latest studies we carried out at my institute on Aedes invasive mosquitoes. It was an amazing chance to discuss mosquito biology and Aedes-borne pathogens with several infectious diseases experts. We also gather ideas to quantify UK habitat suitability for Aedes invasive mosquitoes, such as Aedes coraicus and japonicus. We are definitely going to strengthen our collaboration in the future, as we are going to co-supervise together several PhD students, and we are also going to broaden our collaboration to cover other mosquito species and related transmitted pathogens of European interest, such as West Nile virus.
In the frame of the STSM, I spent a week at the Applied Ecology Research Unit of the Foundation Edmund Mach in Italy. The main purpose of my visit was to become familiar with statistical analysis generally used in mosquito research, particularly the distribution modeling of invasive species. Based on our own data collected since the first emergence of Aedes coraicus in Hungary, we analyzed the dynamics and distribution of the species along with its environmental preferences in our region. This built-up model helps us to understand the dispersal of the species in similar middle-sized European cities with a temperate climate. As a next leap, with several additional colleagues from the AIM-COST network, we analyzed the pan-European genetic patterns of coraicus. That work highlighted the importance of genetic research to understand the dispersal of invasive mosquitoes and the necessity of cooperation without borders. In summary, this short-term visit was a perfect platform for knowledge transfer and the basis for further collaborative work.
Short-term scientific missions is in collaboration with the Friedrich Loeffler Institute with Professor Cornelius Ilagi and Dr. Mandy Schaffer. Our interest, our collaboration was to test the mosquitoes collected in Albania for 3 years, Aedes albopictus, to identify the presence of West Nile virus or any probable other pathogen agents. And we found some positive pools, but we have to wait for the other sequencing methods to identify the lineage of West Nile virus, if it's positive for West Nile virus or for any other other viruses. This is a very good collaboration, and we are thinking to strengthen it in the future. And of course, this STSM was a baseline how to create this network or how to create this collaboration and strengthen it in the future for testing them for West Nile, not for West Nile, for dengue, for chikungunya, to test them for the presence of other filariasis or maybe the potence they have to transmit other infection diseases and their competency they have to transmit it. And we will think to continue this in the future.
Prior to the short-term scientific mission, I conducted intra- and interspecific larval competition experiments at the insectary of the Institute of Tropical Medicine in Antwerp on Culex pipiens, Aedes albopictus, and Aedes japonicus. The adults were raised under selected competition treatments and were transferred from Antwerp to Hamburg to conduct vector competence studies at the Bernhard Nocht Institute for Tropical Medicine. The mosquitoes were sorted, got an infectious blood meal, and infection rate was calculated. A total of 182 Culex pipiens, 32 Aedes albopictus, and 47 Aedes japonicus were analyzed. Infection was successful for Culex pipiens with West Nile virus, for Aedes albopictus with chikungunya virus, but no infection was found for Aedes japonicus with Japanese encephalitis virus. Differential infection rate in response to larval competition treatment was detected. I'm very grateful to AIM-COST for providing me with the opportunity to learn the techniques for vector competence analysis and to build a scientific network at the Bernhard Nocht Institute. We will further collaborate on a joint paper, and I would be thrilled to show our results during the AIM-COST annual meeting in February.
Good morning, this is Mikel, and last August I spent 15 days in Brazil, in the state of Sao Paulo, working on a project called Aedes, a novel tool for the reduction of Aedes aegypti mosquito vectors based on intelligent monitoring. This project is developed by the world's largest pest control company, Rentokil Group, in order to improve health and well-being of humans. Now, our idea is to do a short trial in Spain in order to check if the mosquito traps valid for the collection of Aedes aegypti in Brazil are also compatible for the collection of Aedes albopictus mosquitoes in Spain. If this works, we are planning to start a more comprehensive study in order to detect the arrival of mosquitoes infected with important arbovirus. So now I am in close connection with the people of Brazil, and we are planning the future steps in order to implement this very beautiful and interesting project. Thank you very much.
During my PhD thesis, I worked mainly on culicoides. So after my recruitment, I wanted as an early stage career researcher to strengthen my competencies for other vector groups, in particular mosquitoes. For this purpose, I applied for the STSM, from which I benefited in September 2021. I was welcomed in Applied Zoology and Animal Conservation Research Group at the University of Balearic Islands in Palma de Mallorca, Spain. Indeed, this STSM allowed me to consolidate and strengthen my knowledge and skills in field monitoring, systematics, and taxonomy of mosquito of veterinary and medical interest. Thanks to the competencies acquired during the STSM, I did a study this year on the characterization of mosquito fauna at the Zoological Garden of Rabat, Morocco. The results of this study were presented at the European Society for Vector Ecology Congress, which was held in Sofia, Bulgaria. I was also been able to develop a thesis project about arthropod vectors, including mosquitoes, in dog shelters in four Moroccan regions. Finally, collaboration are underway with the Applied Zoology and Animal Conservation Research Group on different topics, including training and exchange.
Can the invasive tiger mosquito colonize the United Kingdom? Thanks to the short-term scientific mission supported by MCOST, we attempted to answer this question with Dr. Claire Strode of Edgehill University and Dr. Joylan Medlock of Public Health England. We used the mechanistic model DynamAedes to simulate the introduction of tiger mosquito propagules in several points of entry in the UK, and we varied the season and year of introduction to accounting for seasonal and intraannual variability. According to our simulation, even a modest quantity of propagules, roughly 500 eggs, can support the establishment of the species in the southeast part of the country during particularly warm summers. These and other results produced during the short-term scientific mission will be published in a joint publication and help Public Health England in their vector control and management activities. As an early career researcher, I'm grateful to MCOST for granting me the chance to meet and collaborate with Dr. Strode and Dr. Medlock.
Hi everybody. I would like to thank to AIM Cost for short-term scientific mission, which I used to spend 1 month in Germany in CAPS Institute, hosted by Professor Norbert Becker and his experienced team of scientists. During 1 month's stay, I learned how to molecularly analyze Culex pipiens biotypes, but I also gained a lot of practical knowledge by working in a field on monitoring of Aedes japonicus. This knowledge resulted in first detection of this species in Serbia. I would always recommend short-term scientific mission to the scientists because it helps to create great friendships and great collaborations with the people who are dealing with same scientific problems and issues. Thank you, AIM Cost, and thank you, CAPS.
During my PhD program, from which I graduated recently, I had the chance to participate in two short-term scientific missions. On the first stage, I had the opportunity to visit the University of Novi Sad, where under the guidance of Professor Dusan Petric, I had the opportunity to learn about the basic taxonomy of the mosquitoes and also the maintenance of laboratory breed mosquito colonies. On the second stage, I visited the Institute of Hygiene and Tropical Medicine in Lisbon, where under the supervision of Professor João Pinto and Vera Valadas, I was learning basic molecular biology techniques. Both short-term scientific missions highly increased the quality of my scientific work, and I have published several scientific papers upon which my PhD thesis was based on. Last but not least, this program helped me to build a network among experts who work with mosquitoes around Europe. Thank you, AIMCOST, for these opportunities.
Hi, Alex here. I was visiting the Norwegian Institute of Applied Sciences for 2 weeks. I was working with team of Rafi Ahmad. We were doing the nanopore sequencing with the mosquitoes. We were looking for pathogens inside of them. We were using the 16S kit from Nanopore, and we got very interesting results in the end. I learned a lot. I learned how to work with bioinformatics, which was the main point of going to a short scientific mission, and I hope I will be able to transfer the acquired knowledge to the other specialists of vector-borne diseases in Moldova. And now we're working on a short communication, and we hope to publish very nice and interesting results. This was a great opportunity for me, and I hope that this will give me a boost in my working with vector-borne pathogens. And thank you, COST, and thank you for the great opportunity, and see you.
I did my short-term scientific mission in the Bernard Nocht Institute of Tropical Medicine in Hamburg, supervised by Dr. Renke Luchten. I know Renke since our PhD in Culicoides biting midgets. I was involved in Aedes Invasive mosquitoes since the rise of Aedes albopictus in the Balearic Islands. I was always interested in pathogen screenings and host detection, so that's why I went there to do this with the mosquitoes collected in the Balearic Islands. After that, Renke and I applied for a project funded by the government of the Balearic Islands, and we are working now together screening pathogens of mosquitoes collected in rural areas. In parallel, Renke have another project about wing morphometrics, and they are creating a database of wings from mosquitoes collected worldwide, and they take advantage of these mosquitoes from the Balearic Islands for this project. So I hope to see you again in Rome.
The objective of my STSM was to characterize long non-coding RNAs involved in reproduction of Aedes aegypti. In the last decade, it was shown that linker RNAs play a pivotal role in regulating gene expression while they are not conserved among species. This is very exciting because they could constitute species-specific targets for mosquito control. During my PhD, I discovered a linker RNA that modulates fecundity and fertility of Aedes albopictus. Curiously, I identified the homologous genomic region of Aedes aegypti, where there are several linker RNAs as well. My goal was to investigate whether these linker RNAs of Aedes aegypti have a similar effect in fertility and fecundity as in albopictus. Professor Setteling's lab offered me the opportunity to work with Aedes aegypti, a species that is not allowed to rear in Greece. Thanks to the STSM, I obtained promising results regarding the role of those non-coding genes that influence reproduction. My results in Aedes albopictus and Aedes aegypti have a great potential for the development of novel species-specific approaches for mosquito control.
Hello. In September 2020, I was granted for a short-term scientific mission under the Aedes COST Action in Benaki Phytopathological Institute in Athens, Greece. This short-term scientific mission, entitled Vector Control of Aedes albopictus Population Using the Sterile Insect Technique, was for me a key factor to submit a proposal and now to represent the national counterpart in a project supported by the International Atomic Energy Agency for the implementation and validation of SIT. So in October this year, 2022, I successfully conducted the first mark release recapture trial in Portugal. I keep working as a team with several international partners in the improvement of this technique, and I really appreciate the opportunity that was given to me by the Aedes COST that contributed to my scientific career. Thank you.